So hi, I'm Dr. Satya Prakash, and I'll actually be speaking on behalf of both of us. So I'm a co-recipient of this grant, and I'll be leading this project in the lab. I'll just share my slides. So it's really an honor to be invited today and to be a recipient of this grant. Thank you so much to Defeat Duchenne Canada. And it's great to be a new member to this community. I'm really looking forward to getting to know everyone over the next few years. So we are based in Japan, so it's actually morning for us on Friday. So hello from tomorrow. We're at the National Center of Neurology and Psychiatry, Institute of Neuroscience. We're based a little ways outside of Tokyo. Our department is actually the Department of Molecular Therapy, and we collaborate really closely with the National Center Hospital, which sees many individuals living with Duchenne. And this is really great because this allows us to collaborate really closely with the clinicians and the patients and families. So it's a really great environment to be a researcher. And I'll talk a little bit in my presentation later about how these collaborations have been really crucial to our research. So we've already had a bit of a background about DMD and how the gene works, but as we're kind of going to be working on the brain-related disorders of DMD, I wanted to highlight the different variants that are really important. So of course, the full-length dystrophin, or DP427, is expressed in the muscle and plays a really crucial role there, but it's actually also expressed in the brain in these regions that I've highlighted here. And in addition to that, a shorter version of dystrophin called DP140 is highly expressed in these regions as well, specifically during development of the brain, so before birth. And depending on where the mutation in the DMD gene is, you're either going to lose the full-length dystrophin in the brain and, of course, in the muscle, or if you have it downstream of these green regions, you're also going to lose the DP140. So this affects up to around 30% of patients, and losing both the full length and the shorter version of the gene is going to lead to cognitive impairment. So this kind of impairment can be seen as autism spectrum disorder, memory impairment, learning and language disabilities. And of course, for the boys who get DMD, this can be difficult for them to live a full normal life. So really getting to understanding this portion of the disease is what our project is going to be focused on. So just to give a little bit of a background, we know that the full-length dystrophin is expressed at synapses in the brain. So just to give a background on what this is, we know, of course, that neurons are the main cell type that make up the brain. And neurons communicate with each other through electrical signaling. And it's really crucial that this signaling is done in a balanced way. And this coordination is done via synapses. And in the brain, the full-length dystrophin is expressed at the postsynaptic region. It works with a bunch of other proteins in a complex and allows the balanced signaling of these neurons in the regions that are involved in the memory processing, memory, cognitive processing. And it's the imbalance of the signaling in these regions, cerebral cortex, hippocampus, amygdala, which leads to these disorders being seen in boys with DMD. Now, the shorter form of the dystrophin, we know less about. So it's very similar in structure to the full length, which this means that we can't easily distinguish its protein function. Although, looking at previous mouse models, we have been able to find that it probably plays a role in specific type of neuronal synapses called glutamatergic synapses or excitatory synapses. Although we don't know where it is in these synapses exactly, in the presynaptic region, the postsynaptic region, and this makes it difficult to understand how the imbalance is being mediated. Although, as I mentioned, we do know that DP140 is highly expressed during brain development. Its function is really unclear during this time point. Of course, it's very difficult for us to study human brain during brain development directly. So we really need a way of doing this. Until this point, mice have often been used as a model to study behavior in diseases. But as you can see from the schematic diagram, the brains of mice and the brains of humans are actually very, very different. Human brains, of course, are much larger, they have increased complexity and much longer developmental periods. And if you look at this image down here, showing the kind of layers that you see in human brain versus the mouse brain, we have far more layers, up to 6 in the human cortex, and this is partially due to a much longer time period of development in which the cortical regions of the brain is expanded. And this is what allows humans to have higher processing ability, higher cognition that we most associate with humans. And mice are just maybe not the best model in order to study these processes. They do represent certain altered social behavior and elevated fear response, which might be associated with some kind of similar disorders seen in humans. But these processes really can't be perfectly compared, and the developmental processes in the mice are absent in humans. So how can we study these? So our lab for this project is planning to use brain organoids, which are these 3D kind of aggregate models of balls of cells, which are derived from patient stem cells, and they represent specific regions of the human brain. Now, this is actually a really novel technique. It was first published around 10 years ago, but has been really kind of expensive and very labor-intensive experimental technique, which is why now it's only becoming more relevant, and we're hoping to really be one of the first labs in the world to do this. So another thing I want to showcase, which highlights the collaboration we have at the National Hospital here in Tokyo and our lab, is that many volunteers have donated their urine so that we can obtain urine-derived cells as a primary culture from these patients, a really non-invasive method, which is much easier for boys with Duchenne to be able to donate rather than a skin biopsy or taking blood, for example. And from these urine-derived cells, we can perform a method called iPSC reprogramming, which converts these urine-derived cells into stem cells. From these, in the lab, we can make embryonic bodies, which are little balls of stem cells. And by exposing these embryonic stem cells to the right combination of chemicals in a dish, we can force these embryonic bodies to become cerebral organoids, ganglionic eminence organoids, hippocampal organoids, representing that part of the brain. All of these regions, of course, are relevant to the DMD symptoms that we see in the brain. And here I'm just showcasing the organoids that we have differentiated in our lab. So from stem cell point, making the ball of cells, and making these organoids with cortical folding that form spontaneously. We also know, looking at specific markers using antibody staining, that synapses are expressed in this model. So using this technique, using stem cells from somebody who does not have DMD, we looked at the expression of DP140, the shorter isoform, and the longer isoform to follow how the gene is expressed throughout development of these organoids. As I mentioned, we already know from previous data that the DP140 is highly expressed during development, and it has this kind of peak at around 10 weeks post-conception, around 30 weeks before birth. And at the same time, the full-length dystrophin is expressed, but to a lower level. And we were really excited to see that this pattern was replicated in our data as well from the organoid. So the DP140 kind of curves up, matching this little peak. And while the DP427 full length is turned on, it remains stable. So looking at this, we were really excited to see that this could be a great model for studying, using brain organoids to study DMD-relevant brains. So as showcasing a couple of the experiments that we're really excited to do for this project, we'll be using iPSCs as stem cells from patients who have either lost just the full-length dystrophin gene, or both the full-length and the shorter isoform. And we can see that these patients don't have any expression. They're just flatline at 0, replicating what we expect to see. And we would like to examine if synapse formation is affected. So in the lab, by making cerebral organoids and ganglion eminence organoids, we're going to treat the GE organoids with a reporter, which is basically a little piece of DNA which can be delivered to the cells and light up the neurons. We can then cut and fuse these organoids together and then look at whether the transport, excuse me, whether the the axonal growth is affected, the formation of new synapses is affected, and whether this is different based on whether we lose just the full length or both the full length and the shorter version of the isoform. This will allow us to identify potential targets for treating these processes. And this is particularly important because we're looking right down at what's going on during development rather than in adult models, which is mostly what's been studied in mice up to this point. Further on, we're also excited to be doing a single-cell RNA sequencing experiment. So just to give a quick background of this, RNA sequencing is a technique which allows us to take a snapshot of gene expression that's occurring in cells at any one time. So this allows us to see which genes have been switched on, which are downregulated, which are upregulated, and then downstream analysis allows us to perform what we call pathway analysis to understand which processes have been disturbed. By looking at the organoids that have a DMD mutation versus those that are normal, we'll be able to identify which of these processes specifically have been misregulated in our DMD brain organoids. Single cell means that we can look at this gene expression change in individual cells in the organoids. So this is looking at the neurons, astrocytes, and pinning down which genes specifically are affected by the loss of shorter and full-length dystrophin. So finally, this grant will allow us to do really great work in establishing these organoids, establishing the disease pathways which come with the loss of DMD. But of course, as I mentioned, we are the Department of Molecular Therapy, and we're really already thinking about how we can use these models to further improve DMD therapies. So we're already thinking about collaborations with a longtime collaborator of ours, Dr. Matthew Wood at the University of Oxford, and to use these organoids as a preclinical model to rebalance the unbalanced neuronal signaling that we see. So using an exon skipping method, which I'm sure many of you will be aware of, as we've also been involved in many clinical trials, including viltolarsen and brogadursen, which are in phase 3 and 2 of clinical trials at the moment. We're planning to try to perform exon skipping to restore the full-length dystrophin and use this model as a way of restoring the balance that we see in neurons. So thank you so much. I'll just end the talk there.